Hi, I'm Brian Brightbill. In this Kaplan MCAT Science Review video, we're going to be looking at the reactions of the citric acid cycle. On our plate, we'll be looking at the reaction steps, including the substrates, enzymes, and products of each step. We're going to be looking at the net results of the citric acid cycle, including the energy yield. Then we'll be looking at the ways in which the citric acid cycle is regulated. By the end of this video, we want to be comfortable with the steps, results, and regulation of the citric acid cycle. This will allow us to answer questions like the one at the bottom of your screen regarding an activator for the citric acid cycle. The citric acid cycle can be one of the more intimidating topics on the MCAT, in no small part thanks to images like the one on the screen right now. But we're going to break it down into more manageable chunks so that you are more ready to tackle the topic for test day. But before we begin breaking down the steps of the citric acid cycle, let's just take a look at the big picture. The citric acid cycle begins as the acetyl-CoA produced during pyruvate decarboxylation is combined with oxaloacetate. This leads to citrate, and beginning with citrate is the reason this is called the citric acid cycle. And then various other products. Each product is once again used as a substrate in another reaction. The products of the reaction that are not reused are NADH, FADH2, GTP, and carbon dioxide. NADH and FADH2 are energy carriers, whereas GTP is an energy currency that can be converted to ATP for cellular use as an energy source. The rest of the products, since they are recycled, are what make this a cycle. Even though none of these reactions directly require it, the entire process will not occur in the absence of oxygen, as it will lead to NADH and FADH2 building up in the reaction pathway because they're not being consumed in the electron transport chain. Alright, and now that we've taken a look at this big picture, it's time for the fun part. Let's look at these 8 steps in detail. In the first step of the citric acid cycle, acetyl-CoA is combined with oxaloacetate to produce citrate. The acetyl-CoA is directly reacted with oxaloacetate and water. In this process, the acetyl group of acetyl-CoA will combine with the oxaloacetate. Note on the citrate, the acetyl group is now combined with the carbonyl carbon of the oxaloacetate. The CoA has been released. The intermediate of this reaction is citral-CoA, which forms through a condensation reaction between acetyl-CoA and oxaloacetate. The water hydrolyzes the citral-CoA to make citrate. The enzyme citrate synthase drives this reaction forward. A synthase is an enzyme that is able to work without the input of outside energy. This energetically favorable reaction helps the citric acid cycle keep moving forward. In order to continue our journey along the citric acid cycle, the citrate must be isomerized to isocitrate via the enzyme aconitase. In this process, the citrate reacts via the aconitase, and the hydroxyl group and a hydrogen are removed, producing water. That aconitase is a metalloprotein, meaning that it requires iron 2+ as a cofactor. Following this process, cis-acontate is formed, wherein there's a double bond between the carbon that lost the hydroxyl and the carbon that lost the hydrogen group. Then that cis-acontate reacts via aconitase once again to produce D-isocitrate. In this case, the water has been returned. The hydroxyl and the hydrogen groups have been returned to the molecule, but now their positions have switched. So if the only net change from this reaction is a rearrangement of functional groups, then what was the reason to have this reaction occur? This step must happen for oxidative decarboxylation to occur, which will be our next step. In that next step, isocitrate does undergo oxidative decarboxylation, catalyzed by the enzyme isocitrate dehydrogenase. In our first step, isocitrate is oxidized by the isocitrate dehydrogenase. This enzyme is our slowest enzyme, making this the slowest step, therefore the rate-limiting step of the citric acid cycle. The enzyme is a dehydrogenase, meaning that it transfers a hydride ion to an electron acceptor. That means that we are forming NADH or FADH2. 2 hydrogens are removed from the isocitrate. One of them is a hydride and therefore carrying electrons that gets accepted by the NAD+, forming NADH. The other generates a proton. This NADH that is formed is the first energy carrier produced from that original acetyl-CoA molecule. This oxidation leads to our transition state oxalosuccinate. That oxalosuccinate undergoes a decarboxylation reaction. Carbon dioxide is lost from the number 3 carbon. This carbon dioxide that is lost carries with it the first of the 2 carbons brought in from the acetyl-CoA. So this is the first carbon lost from that original molecule. The proton is added to where the carbon dioxide was lost, forming our product alpha-ketoglutarate with a carbonyl on the second carbon. As we move on to the next step, we are forming succinyl-CoA and carbon dioxide and another energy carrier, taking place at the alpha-ketoglutarate dehydrogenase enzyme. Alpha-ketoglutarate reacts at the alpha-ketoglutarate dehydrogenase complex. This complex has similar coenzymes, cofactors, and mechanism to the pyruvate dehydrogenase complex, with which you should already be familiar from pyruvate decarboxylation. In this process, the CoA donates a hydride ion to the NAD+, producing NADH. This is the second NADH to be produced in the citric acid cycle. A carbon dioxide is also driven off in this process, producing succinyl-CoA and a CO2, which represents the last carbon lost from the original acetyl-CoA. Succinyl-CoA is therefore our next substrate. Our next enzyme is succinyl-CoA synthetase. This will produce our only energy currency of the cycle. The succinyl-CoA is broken down by succinyl-CoA synthetase. Being a synthetase, that means it is creating a new covalent bond using energy input. In this case, the bond that is being formed is the phosphorylation of GDP to GTP. The energy source is the hydrolysis of the succinyl-CoA, which is a thioester. The GTP that is produced will transfer its phosphate to an ADP, meaning that the end result is the production of ATP. Coenzyme A is also released here for use at other places around the cycle. The product of this reaction that will serve as the substrate in the next is succinate. It's important to pay attention here to the fact that this is the only place in the citric acid cycle where an energy currency is being directly produced. Here is GTP, though that will quickly interconvert to an ATP. Following succinate formation is fumarate formation, which uses a dehydrogenase enzyme again. This means that another energy carrier will be produced. In this reaction, succinate and the succinate dehydrogenase enzyme produce fumarate. This is the only step of the citric acid cycle that occurs on the inner mitochondrial membrane, while all the other steps occur in the mitochondrial matrix. The reason fumarate formation occurs along the inner mitochondrial membrane is because succinate dehydrogenase is an essential enzyme in complex 2 of the electron transport chain. There, it is involved in oxidizing the FADH2, donating the high-energy electrons to the electron transport chain. In fumarate formation, the 2 hydrogens from succinate are transferred to the FAD, producing FADH2. As in other dehydrogenase reactions, the substrate succinate is oxidized, whereas the FAD, which becomes the energy carrier, is reduced to FADH2. FAD is reduced instead of NAD+ in this reaction because succinate is not a strong enough reducing agent to reduce NAD+. This means you know why there are 2 energy carriers. NADH carries more energy than FADH2. Next in the citric acid cycle is malate formation, driven by the enzyme fumarase. In this step, fumarate is hydrolyzed to produce malate. The hydroxyl group and the hydrogen from water are added to either side of the double bond, breaking that double bond. A small stereochemistry note here: while there are 2 enantiomers possible for malate, only the L-malate forms. And in our final step, we must regenerate the oxaloacetate. In this process, we also produce our final energy carrier. The malate is oxidized by malate dehydrogenase. This produces oxaloacetate. Note the name of this enzyme. This is the same malate dehydrogenase as appears later in aerobic respiration, in the malate aspartate shuttle. But in that shuttle, it can run both forward and backward. In this process, a proton and a hydride ion are liberated from the malate. The hydride ion is accepted by NAD+, making this our third and final NADH produced and fourth energy carrier. At the end of this process, we once again have oxaloacetate in order for the cycle to begin anew. We know that memorizing this isn't the easiest part of preparing the citric acid cycle for test day. So thankfully, Kaplan has an entertaining and memorable mnemonic that will help us get there. The mnemonic is, "Please, can I keep selling seashells for money, officer?" Here, the capital letters stand for the substrates of the citric acid cycle: pyruvate, citrate, isocitrate, ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. With that mnemonic at our disposal, we're now a little more prepared on test day to tackle questions about the substrates of the citric acid cycle. Now, while the MCAT might test you on the substrates and enzymes and steps of the citric acid cycle, they are most likely to test you on the net results. This mostly focuses on the energy yield. So now let's take another look at this exact same process, but change our lens and focus on those results and on the regulation of this process. Before we even reach the citric acid cycle, NADH is produced in pyruvate decarboxylation. Then, as we form alpha-ketoglutarate and succinyl-CoA, two more NADHs are produced. A GTP is produced in succinate formation, an FADH2 in fumarate formation, and one more NADH as we reform the oxaloacetate. As the purpose of cellular respiration is to produce energy, it's important to know in what ways energy is produced during the citric acid cycle and the other parts of aerobic respiration. In pyruvate decarboxylation, which remember comes right before the citric acid cycle in cellular respiration and occurs at the pyruvate dehydrogenase complex, that pyruvate produces NADH, our first energy carrier, and this is where we lose the first carbon from the original 3-carbon pyruvate. In the citric acid cycle, the other 2 carbons are released, 3 NADH are produced, an FADH2 and a GTP. This means for overall ATP production, 4 NADH result in 10 ATP, as there are 2.5 ATP produced per NADH. FADH2 produces 1.5 ATP per FADH2, so that is 1.5 ATP for the one that is produced in the citric acid cycle. And the GTP that is produced is directly converted into ATP by transferring that phosphate group to an ADP. This makes a total of 12.5 ATP per pyruvate. From one 6-carbon glucose, there are 2 3-carbon pyruvates produced, making for 25 ATP per molecule of glucose by the end of the electron transport chain and oxidative phosphorylation. Combining in the original glycolysis that split the glucose, which produced 7 ATP total for the 2 ATP directly produced and 2 NADH, that makes a total of 32 ATP. While this can vary slightly from cell to cell, 30 to 32 is a good range to know for test day. Next, we need to look at how the citric acid cycle is regulated. Our metabolism does not want to be constantly producing as much ATP as possible. We want to be able to control when ATP is being produced through the citric acid cycle and when it is not. Even upstream of the citric acid cycle, at pyruvate decarboxylation, the citric acid cycle can be prevented by preventing this process from occurring. Pyruvate dehydrogenase kinase is activated by high ATP levels, which has an inhibitory effect on the pyruvate dehydrogenase complex. On the other hand, pyruvate dehydrogenase phosphatase is activated by high ADP levels and activates the pyruvate dehydrogenase complex. Next, within the citric acid cycle, there are 3 sites of regulation. The first is at citrate synthase. This step is inhibited by ATP, NADH, succinyl-CoA, and citrate. The next site for regulation is at isocitrate dehydrogenase. In this case, ADP and NAD+ are activators, and ATP and NADH are deactivators. And then, at the alpha-ketoglutarate dehydrogenase enzyme, ADP, NAD+, and calcium are all activators, whereas ATP, NADH, and succinyl-CoA are deactivators. This leads to a few trends that we should notice. ATP and ADP ratios and NADH and NAD+ ratios are the strongest determining factors in whether the cycle is inhibited or activated. This makes sense, as when a cell is metabolically active, ATP and NADH are being converted to ADP and NAD+. But this rise in those levels would activate the enzymes at the checkpoints, leading to increased production of ATP and NADH. Now let's apply the information about the citric acid cycle. Go ahead and answer question number 1. The activator of the citric acid cycle is ADP. The other 3 answer choices are all produced at some point in the citric acid cycle, whether it's as GTP converting to ATP or direct production of NADH and succinyl-CoA. Since this is a negative feedback mechanism controlling the citric acid cycle, products of the cycle act as inhibitors. Now attack this question. The answer to number 2 is C, malate dehydrogenase. We want to know all of the control points of the citric acid cycle. Alpha-ketoglutarate dehydrogenase complex, citrate synthase, and isocitrate dehydrogenase are the 3 control points within the cycle. On test day, we want to know the path of the citric acid cycle, its energy yield, as well as its regulation. For the path of the reaction, we want to be able to follow the various enzymes and substrates around the cycle, describing how one substrate reacts at the enzyme, producing a product that will be the substrate for the next reaction. In order to remember the substrates pyruvate, citrate, isocitrate, ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate, there is the rather entertaining mnemonic, "Please, can I keep selling seashells for money, officer?" As for energy that is produced during respiration, we want to know that 7 ATP come from glycolysis, 25 ATP come from the rest of aerobic respiration per molecule of glucose. This leads to a net yield of 32 ATP per molecule of glucose. As for regulation, we want to know that the citric acid cycle is controlled upstream by regulating the pyruvate dehydrogenase complex. This is generally inhibited by energy and activated by a lack of energy. As for regulation within the cycle, we want to know the 3 checkpoints: citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate glutarate dehydrogenase complex. Each of these is inhibited by ATP and NADH and activated by ADP and NAD+. Thank you for joining me as we've explored one of the more complex processes in the biological world, and congratulations on moving just a little bit closer to the success on the MCAT that you deserve.